Let us first see about the incomplete dominance in mirabilis gelapa. According to Mendel's inheritance, a dominant or a recessive allele of single genes controls the traits is a simple hereditary process. However, the dominance of one allele over the other allele is not complete. Incomplete dominance, always known as partial dominance, identifies such circumstances. Incomplete dominance is a form of intermediate inheritance in which one allele for a specific trait is partially expressed over its paired allele. The result is a third phenotype in which the expressed phenotypic character is a combination of the dominant and recessive phenotypes. Incomplete dominance is also known as partial dominance or semi-dominance. Flower petal color in 4 o'clock plant provides a very good example of incomplete dominance. 4 o'clock plant is the most commonly grown ornamental plant which is the native of America. The 4 o'clock plant gets its common name from the fact that the flower opens up in the late afternoon. When we examine the mirabilis gelapa, there are 3 colors of flowers. One is red, pink and white. This is the result of interaction among the genes. These interactions include incomplete dominance. Here, a red represented by capital R flowered and white represented by small r flowered varieties were crossed, and the F1 progenies all produced pink colors. The genotype is capital R, small r. The F1 progeny is the intermediate between the 2 homozygous parents, that is red and white. This type of inheritance pattern is known as incomplete dominance. When the F1 pink flowered varieties were self-pollinated, they produced red, pink and white flowered plants, and the segregation are in the ratio 1:2:1 in the F2 generation. In this case, red color producing gene is not completely dominant over white color producing gene. The effect of the 2 alleles is more visible. The phenotype is a fine mixture of the 2 alleles. The expressed hybrid is new, but it has no allele of its own. A quantitative effect is present in incomplete dominance. Geneticists can tell that in this case, flowering time of mirabilis gelapa is controlled by the interaction of 2 genes in a single locus, because self-sterilization of capital R, small r plant produces a 1:2:1 ratio in the progenies.